Okay, so welcome to this next video in which we are talking about the adaptive immune system and immunosuppressant drugs. Okay, so we've now finished our discussion of the adaptive immune system and we're going to move on to immunosuppressant drugs, which are drugs which block the adaptive immune response. Okay, so let's talk, before we actually get into the details of the actual drugs, let's talk about basic principles, i.e. if we were just sort of, poised with the question, if we know absolutely nothing about immunosuppressant drugs, and we know all that I've told you about the adaptive immune system, what sort of tactics would you come up with for blocking the adaptive immune system? Okay, well, if you were a child, if you pose this question to a child and the child understood all of the stuff about the adaptive immune system, then you might come up with a very crude method of destroying the adaptive immune system, which is simply everything is based on these lymphocytes. Everything is about T lymphocytes and B lymphocytes. So what if we just got rid of all of the T and B lymphocytes? So if we destroy the lymphocytes, that would completely destroy it would completely destroy the adaptive immune response, destroyed the lymphocytes. That would be a way of killing the adaptive immune response. Now, you might think, oh goodness, how crude, of course there isn't going to be a drug that does that, but we are actually going to see a drug that works literally by killing all of the T and all of the B lymphocytes, and therefore you are not going to be able to initiate an adaptive immune response, because the adaptive immune response, all three of those pathways we saw, they involved T cells and B cells, and if you have no T cells and B cells, then the professional antigen presenting cell isn't going to be able to activate T cells. And those T cells aren't going to be able to activate B cells in the case of the humoral immune response, or in the case of the two-cell mediated responses, we're not going to be able to generate T helper 1 cells or cytotoxic T cells, because there aren't any T cells. Okay, so this very crude strategy for destroying the adaptive immune response is actually going to be a mechanism of action of one of the drugs we're going to see, and we'll look at that later. I'll give you the name of it now so you can know what, because it has quite a terrifying name for quite a terrifying mechanism of action. So it's called alemtuzumab. Okay, and it's a monoclonal antibody. Okay, so we'll look at that in more detail later. Okay, and I should stress that it's only at the moment, I think, used as an immunosuppressant in people with multiple sclerosis. It's actually used more as an anti-cancer chemotherapy. So if you've got a cancer that involves overproliferation of the B or T lymphocytes, which could be a lymphoma, for instance, then alemtuzumab will kill those T and B lymphocytes, and therefore is a very powerful anti-cancer chemotherapy when we have a cancer involving T or B lymphocytes. Okay, so let's go on for more strategies, maybe more tame strategies for how we could destroy the adaptive immune response. Well, another way we could destroy the adaptive immune response is just by stopping cellular proliferation. So if we had drugs which were antiproliferative, would this destroy the adaptive immune response? And my claim is that it would, because if we look at the pathways, okay, so let me just get the pathways back again. So we'll start off with the humoral immune response. Okay, so where did I put the humoral? Okay, so remember, the antigen presenting cell brings its antigen to this naive CD4 positive T cell, okay, and causes activation of that CD4 positive naive T cell, okay, and then the first thing that happens is that differentiates into a T helper 0 cell, but then you get proliferation of the T helper 0 cells to get a whole population of T helper 0 cells. If you blocked proliferation, then you block this from happening, and you'd still only have one T helper 0 cell. Then you'd end up with very few T helper 2 cells. You'd end up with one, okay? In addition, B cell activation, remember, it was completely dependent upon this proliferation of the B cells to form this germinal centre. So again, firstly, you have only got now one T helper 2 cell, so it's very unlikely that the B cell that's found the antigen will actually be able to find a T helper 2 cell with this T cell receptor that's complementary for this antigen fragment. So that's one problem. But also, even if it does somehow, by some miracle, manage to find this one T helper 2 cell with the complementary T cell receptor, it won't actually be able to divide and produce this whole population of B cells which have their B cell receptor based on its B cell receptor. Okay, so again, you're not going to get somatic hypermutation, well, you're not going to get proliferation occurring, and therefore you're not going to get this affinity maturation process occurring, and that means that you're not going to end up producing plasmablasts, which will produce plasma cells. So you're going to kill the humoral adaptive immune response, certainly. Okay. Again, with the cell-mediated response for an intracellular pathogen that was phagosomal, again, it followed the same suit. You activated a CD4 positive naive T cell, and it differentiated into a T helper 0 cell, and then needed to proliferate into T helper into a whole population of T helper 0 cells, which then differentiated into T helper 1 cells. So, if you're blocking proliferation, then those T helper 0 cells aren't going to be able to proliferate, so you're going to end up with just one T helper 1 cell, which is useless pretty much. Okay, you need a huge, great population of them. And finally, for the cell-mediated pathogen where we were actually intracellular, well, within the cytoplasm, you activated a CD8 positive naive T cell, and then what it did was it differentiated into a cytotoxic T cell, proliferated within the lymph node to make a whole population of cytotoxic T cells, which then went into the blood and were recruited to the site of infection, and then went forth and caused apoptosis of infected cells. So if you stop proliferation, you're going to end up with just one cytotoxic T cell, and that's not going to be able to do much. Okay, so again, you're going to hugely weaken this strand of the adaptive immune response. So we've seen, therefore, that all three of the adaptive immune responses will hugely be weakened if we give an antiproliferative drug. Okay, so here is where the immunosuppressants overlap with the anti-cancer chemotherapies, because anti-proliferative drugs are generally used for anti-cancer chemotherapies. Okay, and we'll see one drug, well actually we'll see 3 drugs that work by stopping proliferation. Okay, now 2 of them are drugs which just stop proliferation, or at least their main target for anti-proliferative effects is specifically the B cells and the T cells, and that they don't affect other cells, okay, or at least they don't affect other cells as much. So they're quite nice because they won't have as many side effects, whereas we'll see one drug that just targets all cells in the body and stops proliferation, and that really is an anti-cancer chemotherapy agent. Okay, and that will obviously have quite horrendous side effects, but it is an effective strategy for suppressing the immune system. Okay, and then the other major way that we could stop the immune system is if we somehow blocked T cell activation. So if we stop T cell activation, then that would again block all 3 strands of the adaptive immune system, because remember, in order to get the adaptive immune system working, you need T cells, and you need to activate either a CD4 positive naive T cell in the case of the humoral adaptive immune response, and also the phagosomal cell-mediated immune response, or you need to activate a CD8 positive naive T cell in the case of the intracellular cytoplasmic pathogen. Okay, so if you can block T cell activation, then that again would kill all 3 strands of the adaptive immune system. So these are the 3 main strategies that we're going to take. Now, these ones here, the ones which stop T-cell activation, they're really the more used drugs. This one is very, very a horrible drug, and these are more tend to have more side effects than these ones. So we'll start off with the drugs which stop T-cell activation, but it is fair to say that immunosuppressive agents are quite foul drugs overall. Okay, so none of these drugs are going to be nice drugs to take. Okay, so what we're going to have to look at is the process of T-cell activation again, but now we're going to look at it in a bit more detail than we ever looked at it before. Okay, so remember, let's have our professional antigen presenting cell here. Let's have a reminder of T-cell activation, and we'll look at it with a bit more detail than we've ever seen it before. Okay, so here is our antigen presenting cell, which I will abbreviate as APC, and it has on its surface a major histocompatibility complex. Okay, now we'll say that this is either major histocompatibility class 1 or it's major histocompatibility complex class 2. So I'm not going to specify now which it is, okay? So if it's a humoral immune response, or if it's phagosomal intracellular, it will be MHC class 2. If it's the intracellular cytoplasmic pathogen, it will be MHC class 1. Okay, but we're going to generalize now, and we're going to have this for all T-cell activation, because indeed the process is the same for all T-cell activation, whether it's MHC class 1 or MHC class 2 that we're going to use. Okay, so here is our major histocompatibility complex molecule, and bound on its surface it has an antigen fragment. So let's colour in the antigen fragment in blue again. Okay. And of course the professional antigen presenting cell will also have put co-stimulatory molecules on its surface, which have had their expression activated by the activation of the pattern recognition receptors within the antigen presenting cell. Okay, so this one, let's say, is CD40, and let's say this one here is either B7.1 or B7.2. Okay, so let's colour those in. So CD40 we'll have in orange here. Okay, and B7.1 or B7.2 we'll have in green. Okay, so then we have our naive T cell here. And again, this now we're going to generalise. It's either going to be a CD8 positive naive T cell, if we're presenting it on MHC class 1, or it's going to be a CD4 positive T cell if we are presenting it on MHC class 2. So again, it's either CD8 positive / CD4 positive. So I will put the one that's in front to match the MHC class 1, which is also in front, and the one that's second will be always what we would have in the second case, basically. Okay, so we'll have a CD8 positive or a CD4 positive naive T cell. So basically, if you want to make this specific, what you would do is you'd say, okay, let's pick MHC class 1, cross out MHC class 2, and then in all the other places where we have an option, you'd pick the first one, basically. So here you'd pick CD8 and you'd cross out CD4. Of course, ignore B7.1 and B7.2. That's a different reason that there are two of them there. Okay, so this will then bind to a T cell receptor on the surface of this naive T cell, which is complementary to the antigen fragment. So the professional antigen presenting cell will have had to search long and hard to find this naive T cell that has a complementary T cell receptor to the antigen fragment that it's presenting on its surface. Okay, and it will also need to have the correct CD molecule, i.e. it will need to have the correct it will need to be the correct type of T cell, okay, for the MHC molecule. So this is our T cell receptor, and then of course we know that there will also be a molecule here which binds to the MHC, and if we've got MHC class 1 here, this will be CD8, and if we've got MHC class 2 here, this will be CD4. Okay, and remember, T cells only have one or the other. They never have both of them. They have either CD8 positive, they either have CD8 or they have CD4. At least mature T cells either have CD8 or CD4 and not both. Okay, in the development process of T cells, they can have both for a while. Okay, then this is going to deliver signal 1 to this naive T cell. Okay, so when the T cell receptor binds to the antigen fragment on the MHC molecule, and also the CD8 or CD4 molecule binds to the MHC class 1 or MHC class 2 on the antigen presenting cell, these two things together will deliver what is known as signal 1 to this naive T cell. Okay, then signal 1 will do nothing unless it also receives signal 2, which comes from receptors for these co-stimulatory molecules here that are on the surface of the CD8 positive or CD4 positive naive T cell. Okay, so let's have in pink here CD28, which is the receptor for B7.1 or B7.2. So this is CD28 here. Okay, and let's have in yellow here the receptor for CD40, which is CD40 ligand. Okay, so let's label this up now, CD40L. Okay, and this now delivers what's known as signal 2 to the T cell. Okay, so here comes signal 2. So both CD40 ligand and CD28 need to bind to their respective co-stimulatory molecules on the surface of the antigen presenting cell to deliver signal 2 to the CD8 positive or CD4 positive naive T cell. And now, what happens next? This is what we didn't look at. We didn't look at the details now when we were originally looking at this, but now we need to look at the details. Okay, so the exact same process occurs whether this is a CD4 positive naive T cell or whether it's a CD8 positive naive T cell. Basically, calcium goes up in the cytoplasm of the cell in response to signal 1 and signal 2. Okay, now what does calcium do when it goes up in the cytoplasm of the cell? Well, it's going to bind to a protein that is in the cytoplasm of all cells. Okay, and this protein is known as Calmodulin. So let me just talk to you a little bit about Calmodulin, because it's one of these proteins that people have generally heard of but know absolutely nothing about. So let me just give a little bit of a description of it. So I'll draw a nice little cartoon of it. So Calmodulin is often drawn like this in cartoons. It has these two lobes, the N lobe and the C lobe, okay? And each of these lobes has 2 calcium binding sites. So here are 2 calcium binding sites on the N lobe, and here are 2 calcium binding sites on the C lobe, okay? Now, before calcium is bound to these 4 calcium binding sites, the calmodulin molecule is in this sort of hunched over shape where the 2 lobes are bent back towards one another, and this structure is what's known as apocalmodulin. Okay, and the abbreviation for apocalmodulin is to call it APO, and then the abbreviation for calmodulin is C lowercase a uppercase M, like that, calmodulin. Okay, so what happens is when 4 calcium ions come along, 1 will bind in each of these calcium binding sites, and that changes the conformation of the calmodulin. What happens is it unbends, basically. It elongates back out, like so. So the 2 lobes move apart, basically. Sort of imagine like that, springing back out. And this is once you've got calcium bound to these 4 calcium binding sites. And then what also happens is the exact opposite happens for this linker that links the 2 lobes. So the whole structure has sort of linearized, it's become straighter, whereas the linker between the 2 lobes is going to unlinearize. So usually it's a linear polypeptide, okay? So it's amino acid after amino acid after amino acid. But now it's going to take the structure of an alpha helix. It's going to adopt a secondary protein structure, which is an alpha helix, like so. So it's going to spiral like a spring, okay? And this is once you've got calcium bound in these 4 calcium binding sites. So one's here, one is here, and one is here. So 4 calciums overall bind to the calmodulin, and this creates what is known as a calcium calmodulin complex. Okay, and the abbreviation for a calcium calmodulin complex is to call it a Ca2+ for calcium, okay, and then CAN for calmodulin, and that's the abbreviation for a calcium calmodulin complex, Ca2+ CAN. Okay, so what's going to happen is when the calcium goes up in the cytoplasm of the naive CD8 positive or CD4 positive T cell, after it's received both signal 1 and signal 2, the calcium is going to bind to calmodulin and form calcium calmodulin complexes within the cytoplasm of the cell. And these calcium calmodulin complexes are now going to activate an enzyme known as calcineurin. Okay, so let's put calcineurin here. So there is an enzyme that is sitting in the cytoplasm of your cells which is currently inactive. However, it's going to get a molecule of calcium calmodulin complex binding to it. So here is the calcium calmodulin complex here. Let me just put 4 little green dots to represent the calcium ions. Okay, so here is our calcium calmodulin complex. It's going to come and bind to this enzyme, which is called calcineurin. Okay, and by the way, the abbreviation for calcineurin is very similar to the abbreviation for calmodulin. You abbreviate it as uppercase C, lowercase a, uppercase N now. So CAN is short for calcineurin. Okay, and calcineurin, once it's activated, is what's known as a SerineThreoninephosphatase. So let me explain what it means to be a SerineThreoninephosphatase after I've just coloured in calcineurin in pink here. Okay, so it's going to remove phosphate groups of serine and threonine residues. So let me show you the structure of serine and threonine residues. So I'll draw them as though they're in proteins. Okay, so here is the amino group here. Here is the alpha carbon, so of course this amino group will be bound to the next amino acid along because it's in a protein. Here's the alpha carbon with its hydrogen off it, and here's the carboxyl group, which will then be linked to the amino group of the next amino acid along. Okay, and the R group of a serine amino acid is that you have a methylene group with then an alcohol group on the end like that. Okay, so this is the structure of serine. Okay, and let me now show you the structure of threonine. So here comes threonine. Here's the amino group. Here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group again, which will be linked to the next amino acid along. And then the R group for threonine is that you have a methylene group with an alcohol group still off it, but then you have a methyl group coming off this carbon as well, so it's not really a methylene group. I apologise for that saying that it was. You have a carbon with an alcohol coming off it and then another methyl group there. So this is the structure of threonine. So you can see that effectively they're almost identical. Threonine just has a methyl where serine just has a hydrogen. Okay, so what you can do to both of these amino acids when they're in proteins is you can add phosphate groups onto them. So let me draw a phosphate group here. So a phosphate group consists of a phosphorus atom with an oxygen double bound to it and then 2 alcohol groups coming off it, and then single bond to another oxygen which has acquired an electron ionically and therefore has a negative charge. Okay, so this is a phosphate group. And what you can do is you can attach this phosphate group to the alcohol group on either a serine or a threonine residue. You take the hydrogen off the alcohol group on the amino acid, you take the whole alcohol group off the phosphate group. You combine those together to make water, which is why this is known as a condensation reaction. Okay? And then you bind that oxygen there to the phosphorus atom over here to create what's known as a phosphate ester link, also often called a phosphoester link. Okay, so this is a phosphate ester link. Okay, so I would encourage you to think of it as being similar to a normal ester link, because effectively you've got something very similar to a carboxylic acid group here. You've got well, imagine if this was a carbon here, you'd have a carbonyl group and then an alcohol group, which is very similar to a carboxylic acid group. So let me just draw a carboxylic acid group. This is the carboxylic acid group here. And you can see that this is almost the same, except that you've got a phosphorus there. And effectively, this is just like an ester link. If this had been a carboxylic acid group, i.e. if this phosphorus had been a carbon, then it would be an ester link. But because it's a phosphorus atom there, it's called a phosphate ester link rather than a normal ester link. Okay, so you can add phosphate groups onto serine and threonine residues within proteins, and this can modify the functioning of those proteins. Okay, now calcineurin is not an enzyme which sticks phosphate groups onto serine and threonine residues. And I should say that generally the enzymes which do stick phosphate groups onto serine and threonine residues, which are known as serine threonine kinases, they don't actually just take an individual phosphate group like this and stick it on. Instead, they usually take an ATP molecule, rip off one of its phosphate groups, and then stick that onto the alcohol group of the serine or threonine residue. And the energy that they get from hydrolyzing the ATP drives the reaction. Okay, so calcineurin, however, is not a serine threonine kinase. It's what's known as a serine threonine, not forward slash, not a dash, serine threonine phosphatase. So it's going to go around removing phosphate groups from serine and threonine residues. Okay, and we'll see the importance of this in the next video.